So I'm going to give a talk on how to store MEGANv5 datasets on a web server today. So it's not a fancy classification tool or alignment or something, but it's basically the answer to what you do with if you create 50 metagenomic samples, all of them like 50 gigabytes big, and you want to share them with a friend or with somebody else. You don't want to ship all of the data directly to them. You want to somehow ship them in a format which MEGAN can read, but you don't need to send it all. You can send it just by having a web server without duplicating data. But first of all, I also work on mouse metagenomics, and metagenomics is like quotation marks because it's 16S up to now. Mouse metagenomics and colitis. So it is together with Daniel, Julia Frick and Isabel Flade from Tübingen University Clinic, and they basically had the idea before the project started that, well, I mean, colitis is a disease in the gut where you have problems with digestion and diarrhea and inflammation in the bowel. And the question is, who is the cause? Well, we have a mouse model, so is a mouse the cause? Is one bacteria the cause? Is one bacteria which somehow treats another bacteria bad the cause, which then there? Or is it a molecule or a protein which causes colitis? So the project started, and Julia and Isabel had already the idea that somehow if we take two genetically identical mice, in that case it's RAC1 minus mice, it means they cannot develop an adaptive immune system, and you have one type of gut microbiota and another type of gut microbiota, so it's a color which distinguishes two the same mice but two different gut microbiota. And if you then add T cells, one of them will develop colitis, question mark, and one of them will stay the way it was. So this year, beginning of this year, the paper was published which showed that actually I can remove the question marks there. It's true that there is a gut microbiome which inhibits the generation of colitis, and there's one if you add T cells, it will develop colitis after a couple of weeks. So this is the same mice as before, and there is some method which is called culture, where you have a look how much in the feces is Bacteroidetes and how much is Enterobacteriaceae. And it showed that they knew already before, so they put into the gut microbiota that one of them more Bacteroidetes and in the other one more Enterobacteriaceae, and of course in less Bacteroidetes in the other one and less Enterobacteriaceae in the other one. And that one, which is called endo low and endotoxicity low, it means didn't develop colitis, and the one with endotoxicity high, apparently causes the Enterobacteriaceae, develop colitis. And if you do sequencing, it was 454 sequencing, you can see that there is definitely a big difference between these both, especially in Bacteroidetes, but also in Firmicutes, which apparently fill up on what Bacteroidetes is missing, but also like this little bit which is the Escherichia Enterobacteriaceae, Proteobacteria is just a level higher, and here it was far under the detection level, not far under the detection level, but I merged a couple of samples together and it was still not findable. Okay, so if you can show that one gut microbiota can inhibit the development of colitis, you want to know, can you take the ones which are actually deemed to develop colitis after a while, can you somehow make them that they don't develop colitis, and the ones which actually stay healthy, can you make them develop colitis? with a similar treatment. So both of them got at day 0 antibiotics. This one got a little different antibiotics than this one, because here you actually wanted to kill the bacteroidetes, and here you just wanted to make a broadband antibiotica in there. And then you added some E. coli, one mutated and one wild type. And you could see that they actually, after a while, that one developed colitis and that one didn't develop colitis. We could even boil it down not only to like the strain of E. coli, but also to one lipopolysaccharide, which is in the LPS. So it was tested if they just put 2 different types of LPS into these 2 different mice instead of this E. coli, the same result was there. Yeah. So our job was there to analyze the data. Well, over the past 3 years, they sent us a lot of CDs with 454 sequencing data, 250 samples. After pre-processing, and that was deleted like more than 50% of the reads, we got 1.2 million reads, so it means 5,000 reads per sample. There's just a problem. We have samples with 500 reads and we have samples with 15,000 reads, because in between we changed the sequencing facility. So it's not that comparable as you actually would like to have it. So what do you do if you have 257 samples of 16S? Either you do some cluster-based analysis, like with QIIME or with Mother. With QIIME you can also use the tools which are in there and not only QIIME, or some other ways. So I usually do a pre-processing with some other tool, import it into QIIME to make the figures and the beta diversity. And you go through this pipeline, and at the end you iterate, because you don't like what you see and you have to start over again. You have to remove samples, you have to add samples to other selections, you have to merge, and so on. And the same thing, or a very similar thing, you can also do alignment based. So you align or classify the reads which you have. Of course I have to promote MALT. So the figures which you see next with the alignment is all done with MALT. You import the result into MEGAN. You visualize the taxonomic composition. You make beta diversity, PCA plots with it, alpha diversity. There's in MEGAN, I think, only rarefaction plots in there. And then you iterate again. Iterating can be painful. Of course, we are talking here only about 1.2 million reads, so it's teeny tiny. But if you go to metagenomics, we also have samples on the web server which have altogether 250 million reads, and it's like 300 gigabytes of space. So iterating through that kind of data in terms of merging, removing, adding samples, if you're working on the file system level, is kind of painful. So the idea was to develop a software where MEGAN can communicate with, because even though the files which MEGAN produces are really big, I mean, they contain a lot of data, the data which is actually transferred to MEGAN is so selective that it can be boiled down to a couple kilobytes every time you click on a button to open a tree, or that you want to see the alignment directly. So as long as you only want to do that, it's only a couple of KB which you have to transfer. So I'm just showing here, like, for example, if I do the reads, comparing them against MALT, and then I put them onto the tree, then I create a file with a MEGAN file, just to show you. Of course, you can also do functional analysis, but because I will only show pictures of 16S, it doesn't make sense to show it now. I also have only 20 minutes time, so better skip it. If you have a couple of samples, these are 12 samples. It's actually permafrost samples. If you decide to compare them, you just open all 12 of them, hit the compare button, and decide and get such such a plot, and from such a compare you can also create a PCA plot of them. So these are 12 samples in a PCA plot, and we will see that a couple more times in the next minutes. So as I said, MEGAN needs very little data in order to work with the data. So why not put all the data which MEGAN produces somehow on a big web server? And MEGAN can communicate with the web server via HTTP. And in the web server we have, like, for example, a Tomcat, and the software which I wrote is only in web archive. So it's a Java program, and we put all the sequence alignments into a big CouchDB, and all the classifications and the metadata we just dump into a Postgres database, and every time somebody wants something from MEGAN, you can just go in here, have your user data filled in, will be checked, and then you just get the classifications and have a look, and selectively it will work very fast. So, and this is a tool which is built into MEGAN, and you can basically, on the left side you can see the 3 running servers which we have at the moment, and on the right side you can see something like a file system structure. It's a tree, so in that case it's the number of the sample, and you can select them, and you can decide what to do with the samples. So there's a couple of options. Either you open them as individual samples, and you get 3 different samples. Maybe you want to look for one specific bacterium, want to have a look at the alignments in there. Or if you want to open the things as a compare, you can also do that with MEGAN server. Just collect the tiny bits of the classifications, puts them into a compare data format, and gives it to MEGAN. Or the third option is to merge a couple of samples together, either in memory or on the server, then you store it there. If you just do it in memory, it's for free. So this is a merge of 3 samples. You can see it looks like that one. It's just a merge of the samples. Why do you want to do it? Maybe you're looking for something which is there just 1 in a million, and you want to find that protein or that species somehow, and that's why you need to merge. Or you want to, instead of comparing a number of samples with each other, you want to compare scenarios. For example, sick versus healthy. I will show that in a couple of minutes. So what I did is I opened 257 samples in a compare. Took a while with my laptop. Then I created a PCA, and I got that one. PCA only helps you if you are able to somehow know what the dots stand for. So that's why we collected, or better said, Isabel from a medical department collected metadata, 29 different fields of metadata for the 16S samples, for 257 samples. And if you color them, for example, like that, you can see something like a nice clustering. The bad thing, it's from the animal facility, because we had 2 different animal facilities. The good thing is, and if you color them by experiment, so they changed not during experiments, animal facility, but in between experiments, you can also see a nice clustering here. Here, not here, but for example, if you now want to have a look at only this experiment, what would you do? There are 60 samples. Do you want to open them all by yourself, find them somewhere in the hard disk? No, you want to use metadata for that, because metadata is stored on the server. You have a Boolean expression parser, say experiment number 4, you know that before what you're looking for. If you hover, if you check it on the sample viewer, which was here, you know for what experiment you're looking for, and then click on the Analyze button and open only the samples which originate from experiment 4. Now I colored it in from the time points. So brown is a time point where they just got a couple of days before antibiotics. You can see the result on the broadband antibiotics, and you can see the result of the antibiotics which were only removing bacteroidetes. This is a couple of weeks later where you start seeing the result, the beginning of the infection sum, and this is later where this group actually got sick, this one stayed healthy, and this one, the experiment didn't work. You can see that one. So it's actually that one and that one belong together, that one and that one belong together. So the next step is then, if you want to dig deeper into the analysis, is to say, hey, I actually want to take all of it from experiment 4. I want to take all healthy ones, and I want to take them which have been sampled when they were actually supposed to be sick or healthy. So I take all of them which were healthy. You can see it here. You take all which were sick, and you compare them to each other. And then you're a little bit disappointed because you expected that the difference would be a bit bigger. But apparently there's a difference, and Isabel from the medical department explained that to me, is enough to make them sick, or it is enough to cause the colitis. Okay, and with that I'm nearly done. So I went really fast through the presentation. I just want to tell why you should use it. Storage solution for a larger project. If you have 250 samples, you don't want to share this data with like sending it to other people, or if you update your data once in a while, you can delete the old data from the server, can put the new data. For example, if a new silver version comes out, you want to update your data, you want to remove the old one without sending it to all the people which used the data before. You can access it via MEGANv5, which has a very nice graphical user interface, and via HTTP, so firewall shouldn't be any problem. Sharing is simple. Selecting with metadata is important. You can project in any way, like one-to-one, n-to-one, or compare. And merging comes for free, absolutely for free. And that's it. Thank you for your attention.
We have time for some questions.
Okay. Yeah, very nice. Maybe just two comments. So we actually find the same, and I think it's a key insight on the subsets. You said in the beginning that we actually usually only transfer very small subsets and then do analysis on this.
Yeah.
I believe that's one of the key insights for analyzing many of our data sets. And the second is also actually a comment, and I'm using a soapbox now. You're my soapbox. I apologize. If we do metadata, we have an international standard group, and George and I are members of that. It's called the Genome Standards Consortium, the Genomic Standards Consortium. And we are really imploring everybody, if you encode metadata, please adhere to the standards. And if there are no standards for your field, you can actually work with us, and we will help you sort of integrate standards. The standards are very onerous. There's 12 fields. One of them is your email. Your first and last name are among the other minimal standards. But there is ways of capturing even more data. And it's really, I mean, you've shown very nicely how adding the metadata helps you actually understand your data, right? It's really the metadata that lets you understand all the sequence data. So if we as a group and all of our friends sort of started using this metadata for real, that would add tremendous value.
We don't have like a standard on what. Of course, you can put all the fields which you were talking about also to there. We didn't do it, but I mean, we don't say you have to put that. Yeah.
Can I just ask you, Volker? So you're not talking about a format. You're talking about an actual just minimum number of tags that everybody should.
We actually have a bunch of suggested formats, but in the end, we can transform between the formats. It's really as simple as the community got together a couple of years ago and agreed on a minimum number of fields, and now there's actually consortia forming for various other areas. We just published something on built environment standards, and there are some marine standards. That can be additional things that you, with your medical friends, for example, can decide on. And actually, we're working in the same area, so we could get together and start talking. What additional things are important for, GI tract disease? But if we all sort of got together as a community, the code providers, if you will, we can make a lot better tools that are allowing interchange. So if you use the standards, you can use Chime to color all your plots, if that is interesting to you. And you can certainly use mgraph right now. I believe Mother supports it as well. It would be lovely if we could sort of stick together as like a sort of group, really.
Or in Chime, you just have a table with metadata, like it's
But
basically a metric.
that's actually GSC compliant.
Yeah?
Yeah.
We use this.
That is the same one.
And you guys are GSC compliant. Lovely!
I tried with the GSC, and I tried to use this GCDML language importing into Java, and it was just too painful to go.
Let's
To
talk
go on
offline.
with
And I agree.
So, Hans, if somebody wanted to install their own instance of Megan server, how easy is that? And can they just contact you to?
Yeah. I mean, it's basically we don't take care of the web server. The only thing we take care about is like what is in here and the Tomcat. So it's a Java servlet engine, and our program is written in Java. So you just get a little file which is like 10 megabytes. You put it into your web server and install CouchDB and Postgres, and the rest happens by itself. So if you want to use it, you can contact me.
Any further questions or comments? If not, thank you.